Hello students, welcome to another module. In this particular module, we will be talking about early land plants or early vascular plants. And here we will be talking about Cooksonia and Rhynia. First we will be talking about Cooksonia. Cooksonia is the earliest known vascular plant. It has been found from Middle to Upper Silurian to Lower Devonian, and the places from where it is being found are Ireland, Scotland, Germany, Russia, North Africa and North America. The plant has been dichotomously branched with an aerial axis which are terminated into an sporangia. There is a basal part of this plant. The sporangia exhibit various shape from round to reniform and contains isospores with trilate aperture. The various species of Cooksonia can be classified or characterized based on the shape of sporangia and its subtending axis. Because of the variability in the form of sporangia, sometimes it is very difficult to identify the species. Here we will be talking about 5 different species of Cooksonia based on its sporangial type. The first one is Cooksonia pertoni. The sporangia of this plant are much broader than high. They are often so flat that they look plate-like. The subtending stalk is strongly widening, whereby the bearing part is as wide as the sporangia. Here is the particular pictures of pertoni. You can see the sporangia, it takes the shape of a plate. Second, we will be talking about Cooksonia cambrensis. The sporangia here are globose or elliptical, and it is bearing on the stalks which are not or hardly thickening below the sporangia. You can see the picture here. Then we will be talking about the Cooksonia calendonica or Arborlenia calendonia. The sporangia are variably shaped but in most cases wider than high or sometimes even kidney shaped. At the upper side, a coaly border can be seen in some cases. Then we come to Cooksonia hemispherica. The shape of the sporangia is semi-globose, globose or elliptical. The stalk is wide below the sporangium and the sporangium is at most about 3 times as wide as the top of the stalk. Sporangia close to the bifurcation are mostly nicely round, while sporangia at longer stalk are more irregularly shaped. Then we come to the Cooksonia paranensis. This species has been described by Gerini in the year 2001 on the basis of 100 specimens from the Lower Devonian of Brazil. The plant has a plate-shaped or bowl-shaped sporangia and it is shrunken within the axis. Sporangia and stalk together are trumpet-shaped. Here we are seeing the different types of sporangia. The figures are given here. Here I have given a picture of the fossil. You can easily see that how this Cooksonia look like when it is present in its fossilized form. And this is the description of this particular Cooksonia. It is an erect plant with naked isotomously branching axis. It is 0.3 to 1.1 millimeter wide. So it is quite a minute plant. So here it says that the uniform size of many specimens described so far favors that Cooksonia is a small plant. But more recently, a larger specimen have been described. So this concept is now no longer valid. Now there we say that Cooksonia is considered as the oldest vascular plant, but the assumption is equivocal. When the genus was originally described, the axis was not terminated into a sporangia. Presence of a central stand composed of specialized cell was only observed in isolated axis which lacks the sporangia. Axes containing terminal sporangia of the Coccinea type have been found to possess thickened walls, but there is no vascular elements. In addition to axes lacking clearly defined vascular tissue, specimens of Coccinea have also been reported with a variety of sporangial morphologies, variously ornamented spores, the axis with or without stomata, and various forms of branching. Now, there is a controversy amongst the various scientists regarding the position of Coccinea, that in which group this Coccinea should be placed. Edward and Edward placed Coccinea in a group they refer to as Rhinioptoides. Taylor terms this group Coccinoides, defined as small plants with terminal sporangia borne on narrow axes that lack true tracheids. Other authors simply include Coccinea-like axes in the Eutrichiophytes. All of this interpretation underscore that Coccinea are presently understood as a highly artificial group of plants that existed during the late Silurian to early Devonian, and that may include forms that are ancestral to either bryophyte or vascular plant, or maybe possibly both of them. Here is the phylogeny of this particular Coccinea genus. Actually, this is a very newer concept where you can see these 5 different species of Coccinea are placed together, and this genus is said to be polyphyletic, and these some are unresolved and have been placed between the Euphyllophytes and Lycophytes. The poorly preserved Coccinea hemisphaerisca is placed as the most basal tachyophyte. 2 other species, Caryopyralites and Caledonica, are placed in stem group of the Lycophytes. A second cladistic analysis was carried out using only 3 best preserved samples, and thus was known. The position of Coccinea caledonica was confirmed, but Pertoni and Parensis now formed a single clade more clearly related to the Lycophytes and Euphyllophytes. So this controversy is still going on regarding the exact placement of this Coccinea. Now we will be talking about Rhynia. Before that, we should know the exact systemic position of Rhynia. Firstly, it belongs to the division Tracheophytes, class Rhinopsida, order Rhynialis, family Rhyniaceae, and hence it's the genus Rhynia. These are extinct vascular plant. So these are no longer found, and these are found from Devonian to Paleozoic age. And 2 species of Rhynia have been confirmed, one known as the Rhynia major and the other is Rhynia guinevogni. So here are the certain characteristics of this Rhynia. The plant body is differentiated into a subterranean rhizome with an abruptly upright photosynthetic aerial shoots. You can see here that this is the rhizome, and below the rhizome there is this tuft of unicellular rhizoids. No true root are found in this particular genus, and this aerial shoot is being abruptly from this rhizome, which is further terminated into a sporangia, and these are dichotomously branched. In regarding this external morphology, this Rhynia major and Rhynia guinevogni can be easily be separated. In case of Rhynia major, the aerial shoots were smooth, But in the second cases, you can see some adventitious branches are present along with the normal aerial shoot. It is being reported that this adventitious branches helps in vegetative propagation. We will be talking about the various anatomical details now. More or less, the aerial shoots and rhizome are almost similar. So, the TS of the aerial shoot can be differentiated into the epidermis, the cortex and the stele. The epidermis is the outermost layer and it is formed of single layered of cell. Outside the epidermis is a one cell thick layer of cuticle. So, this epidermis is cuticularized. In the aerial shoot, this epidermal layer is interrupted by the presence of stomata, but stomata is absent in the rhizome. Coming to the cortex, the epidermis is followed by the cortex. Here also, the cortex forms the major portion of the rhizome. The cortex is differentiated into an outer cortex and an inner cortex. The outer cortex is 1 to 4 layer thick and are thin walled. No intercellular space is present there. But in case of the inner cortex, large intercellular spaces are present and the cells are present with chloroplast. And it is thought that it is the chief photosynthetic region of the plant. In this particular anatomy of the rhizome, we see that there is no endodermis or pericycle. In this picture, you can easily see what I was just talking about. At this outermost layer is a single layer of cuticle followed by the epidermis. After the epidermis lies the cortex. The outer cortex is formed of large, compactly arranged cell. Below the outer cortex is the inner cortex. This inner cortex is formed of loosely arranged cells. The size of the cells are little smaller than the outer cortex and large intercellular spaces are easily seen. At the middle of the cortex lies the stele. The stele is protostele or haplostele in nature, and the middle central core of xylem is being surrounded by phloem. But there is no sieve plates in phloem. Here I have given a structure of the stomata. I have told that in case of aerial shoot, the stomata may be present, and in the epidermal layer, this stomata is being present. This is just an enlarged view of the same. This is the guard cell, and here we can get the stomatal pore. Now we talked about Rhynia major. Rhynia major is now transferred into a new genus. There are certain reasons for this, and this new genus is known as Aglophyton major. This Aglophyton major is now reconstructed as a plant. It is 18 centimeter tall and it consists of a system of naked stomatiferous cells and a loosely lying on the substrate surface function as the rhizomes. We are saying that this Rhynia are the vascular plant, but it is not a vascular plant having a different branching pattern and no central xylem strand. That is, here no central xylem strand is present. The erect dichotomizing branches parted at a wider angle and arose from the prostate rhizome bearing axis. The central conducting strand comprised of elongated thin-walled cells without having the characteristic second thickening of vascular plant. The sporangia are terminal and fusiform in shape and it consists of isospore. So, some says that Aglophyton major was a non-vascular plant with pteridophytic life cycle. So, here we will be talking about the various scientists and their work and what were the reasons that they created this particular genus Aglophyton major. Firstly, I told that there is no discrete secondary wall thickenings on the conducting cells. Some refers that this absence of thickening can be due to poor preservation. Some says that agglophyton measure was actually something quite different. The central conducting cell or the central conducting strand consists of three regions. The outer zone consisting of the phloem and is made up of thin-walled elongated cell with oblique C-shaped end cell. The remainder of the central strand consists of two zones of opaque cells that are initially thought to represent tracheids. The cells in the outer zone are circular in transverse section. In longitudinal section again, it forms a reticulate patterning on their walls, and this may be appeared due to wall degradation or silica crystallization. Cells in the center of the strand are uniformly thin-walled and more angular in cross section. So, Edward concluded that the cells of the conducting strand of agglophyton are more similar to the lepidioids and hydroids cells found in certain bryophytes than to the sieve cells and tracheids of vascular plants. So, this is the transverse section of agglophyton axis showing central strand and containing arbuscule. There is a very peculiarity in this particular structure. It has the axis consisting of parenchymatous cortex, inner cortex, outer cortex. Now this outer cortex is densely packed. It has elongated cell with narrow intercellular spaces, and the outer cortex have hypodermal tissues. But between the outer and the inner cortex, there is a well differentiated region of cortical tissues. In this particular cortical tissue, there is an endomycorrhizal fungus known as glomitis, which forms the intracellular arbuscules. One of the earliest theories suggests that agglophyton measure represented the sporophyte and the Rhynia gynae vogani is a free-living gametophytic phase of a single plant. The idea gained support from the homologous theory of the alternation of generations, since there were no specimen of the latter known to possess sporangia at the time. The theory was abandoned since Rhynia gynae vogani was subsequently shown to possess terminal sporangia, and agglophyton measure is not known to be somewhat structurally different from a vascular plant. Another theory suggested that protrusions near the base of Rhynia gynae vogani axis represented archegonia. According to this hypothesis, the rhizomatous portion of the plant represented the gametophytic phase, and the aerial upright axis bearing sporangia consisted of the sporophyte. Such a situation might be viewed as evidence from the antithetic theory and parallels to the life cycle of the modern bryophytes, where the sporophytic phase is permanently parasitic on the gametophytic one. Although this hypothesis cannot be totally discontinued, to date there is little structural evidence in the form of transitional zone which should represent at the base of all Rhynia gynae vogani plants. So now we will be talking of the reproduction. So here it is the structure. You can see that most of this axis is being terminated into a sporangia. So each axis is terminated into a sporangia. The spores produced in the sporangia are all alike, that is, it is homosporous. Each sporangia is oval or slightly cylindrical with a little greater diameter than of its aerial branch. Now let us take a look of the longitudinal section of this sporangia. The sporangia is a 5-cell thick one. That is in the sporangia, the layers that cover the sporangia consist of 5 layers. The outermost layer is 1 cell thick and it is the cuticulized epidermis. It is followed by a 3-cell thick middle layers of thin-walled cells. Ultimately, in the innermost layer lies the tapetum, which is 1 cell thick. The wall was surrounding a spacious sporogenial cavity which was without columella and contained large number of spores. The spores are same in size and measured up to 60 micron in diameter. This is the particular structure. You can see this is the spores that are present here. This is an enlarged view. This is the sporogenial wall. Our tapetum wall is present. This is actually the fossil in which this particular sporangia remains embedded. The spores here, we can see the spores which have a trilete marked. Now, how the dehiscence of the spores takes place, there is no special mechanism and maybe the liberation of spore takes place just by disintegration of the sporangial wall. Now coming to the gametophytic stage. In Rhynia, we see that there are 2 gametophytic plants, the Lionophyton rhyniensis and Sciadophyton. Both have been described from the Rhynia chart. Lionophyton rhyniensis had a rhizome with aerial axis. This aerial axis has a terminal bowl-shaped lobed gametangiophore bearing antheridia on the inner face of the gametangiophore lobe and the groups of archegonia at the central position within the bowl. It was suggested that Horneophyton lingeri might have been corresponding sporophyte plant of Lionophyton based on their lobed morphology and having common association in the Rhynia chart. However, the gametophore of Lionophyton was also a reminiscent of Aglophyton major on the basis of their central conducting stand as well as having stomata and cuticle. The other gametophyte is called the Sciadophyton, and this Sciadophyton is again very much similar with the Lionophyton. The Lionophyton has a variable number of vertical terete axes that occasionally dichotomize. The central conducting tissues contain some amount of tracheids. So this particular axis terminates into a club-shaped gametangiophore containing antheridia and archegonia. The existence of such gametophytes in early vascular plants supports the theory of isomorphic alteration of generation in which both sporophyte and gametophyte are isomorphic, photosynthetic and contains tracheids. Now coming to the total life cycle. Now spore germination produced a prostate gametophytic thallus composed of dichotomously branched terete axes. These are photosynthetic and contain vascular tissue. As development proceeds, some axes grow upright to become the gametophores, terminated by a bowl-shaped structure bearing the archegonia and the antheridia as in Sciadophyton. Fertilization is initiated by production of sporophytic plant having dichotomous branch. As the sporophyte matures, they produce erect branches with sporangia. Here we have given the various type of gametophytic phase. See, this is the Sciadophyton, this is the gametophores. This is just the enlarged tip of the gametophore. This is the enlarged tip. We told that in case of Lionophyton, that is a cup-shaped structure. And in this cup-shaped structure, the spores are being present, the gametes are being present. Here you can easily see the antheridium and the archegonium. The archegonium is relatively larger in size and antheridium are smaller, and they are present at the tip of the gametophore cup, while archegonium are present a bit down. So this is just a suggested life history or a suggested life cycle. What we see here a mature sporophyte is present. Each bears the sporangia, and in this sporangia two different types of spores are produced. One is forming the male and one is forming the female. So you can see how the spore is slowly developing and how this particular gametophyte is being produced. Similar case with the male also, the spores slowly germinate. So for the better understanding, different color has been used to designate the spores. As a result, this life cycle continues and the blue develops anthridiophores and the orange develops the archegoniophores containing the male and the female gametophyte respectively. So what we can conclude, in all the cases, these sporangia are terminal in position on aerial branches, thus exposing their spores to wider distribution by air current. The wall of the eusporangia are many-celled thick and is provided because why this is thick? Because it provides protection to the spores. The latter, when matured, a thick wall also protect them from desiccation and also from any mechanical injury. The egg cell of the female structure are also protected by the surrounding gametophytic tissues.